Hello, future biologists. Welcome back to Cosmic Edutech by Dr. Priyanka, where we discuss everything from biology to biotechnology. Today I'm here with very simple but yet confusing, but again, it's a very important concept in biology, that is attenuation in trp Operon. So many a time students find it very confusing to understand it and remember it. And the problem is during the exam, you tend to forget what is this finally. So don't worry. By the end of this video, you will be pro with this concept because I'm going to share some very easy tricks and easy explanation so that you can remember this attenuation in trp Operon forever and ever. So let's get started with this trp Operon. So please see this image and just fix it in your mind because it's showing an E. coli and it's showing a stop sign for the DNA. So let's see what is this all about. So let's get started with this attenuation in trp Operon. So we already know how does repression works in trp Operon. So we know it is going to turn off the switch for trip synthesis or a trp Operon. But there is yet another smart way or a precise mechanism which is used by bacteria, that is the attenuation. So attenuation is like a smart way or the fine-tuned gene expression mechanisms, okay, based on how much tryptophan is available. So if you remember in olden days, people used to play FM radio. Okay, so Akashvani and all. But sometimes what is to happen is even if the frequency was set properly, there used to be some noise. Okay, so what could be done? People used to fine tune it so that there is enhancement in the quality of the music that was produced and also there was reduction in the noise. So similar way, attenuation is like the fine tuning mechanism which makes the trp Operon work in very precise and crystal clear way. So attenuation happens only in prokaryotes because transcription and translation they work simultaneously. So that is one condition which is required for attenuation to help or attenuation to work. Now let's see why attenuation is needed. We already have repression. Why complicate our syllabus, right? But no, though it is a complication of our syllabus, but the prokaryotes definitely need this mechanism. So repression can work the best when the tryptophan is either very high or very low. So that time repression can work in a very precise manner. But what about when the tryptophan is present in moderate levels, so moderate amounts? So when tryptophan is present in moderately high, repression alone cannot help the process of trp Operon. So attenuation is something that is going to come and take over. So its strategy is like fine control, no wastage of energy. Because if you recall the previous videos, we had seen that Operon is a smart mechanism which is going to help the bacteria to take the decision what to do, and so that there is no wastage of energy at all. The resources can be saved, the energy can be saved. So that is where your attenuation is going to come into picture. So next, now let us try to understand one very important feature which is present in trp Operon. That is the presence of leader sequence which was not present in the lac Operon. Now the strip L, that is the leader sequence which is present, it is 162 nucleotide leader sequence. And if you see, it has 4 regions. Region 1, 2, 3, 4. So here I have taken an image from internet. Again, let me tell you, it is not my image. I have taken it from the internet. So this, it codes for leader peptide with 2 trip codons and it is located just before the trip E. And there is folding of this particular sequence that is going to allow the transcription to go ahead or to stop. whether to terminate it or allow it. So that decision is going to be taken by the folding of the RNA in the folding of this region in the leader sequence. The play of these four regions, 1, 2, 3, 4, how they are going to form the pairs. That is going to decide what is going to happen the next. So now let us see the attenuator, meaning how the fine tuning is going to happen. So based on the RNA secondary structures, that is the folding, okay, if the region 1 and 2 is forming the complementary loops, it is going to form a pause loop. Then next, if the region 2 and 3, they are forming a hairpin loop, then it is going to form something called as the anti-terminator loop. Anti-terminator meaning it is going to allow the process to happen. And the last one, that is the 3 and 4. If these are formed, it is going to form the terminator loop. That means formation of this particular loop, it is not going to allow the RNA polymerase to move ahead, and as a result the process is going to stop abruptly. So this is how the play of these four regions in the leader sequence is going to play a major role in the attenuation process. So if tryptophan is high, that means the prokaryote or the E. coli already knows that it has enough of resource. It need not go ahead with the transcription, because why waste energy? I already have enough of tryptophan. I can be very happy with it. So the ribosome, what it is going to do is, it is going to quickly translate the leader sequence, okay, leader peptide, and it is going to cover the region 2. Once it is forming the region 2, okay, it is covering the region 2, what is going to happen? Obviously, these 3 and 4 regions will be free. So they are going to form a loop, and as a result, the RNA, because it is a terminator loop, right? So RNA polymerase cannot move ahead and it is going to fall off, and as a result, there is no transcription that is going to happen. So as a result, there will be no tryptophan operon functional when there is high tryptophan, right? So next thing, what happens when there is low tryptophan? So if tryptophan is low, that means the cell definitely is looking for tryptophan and it is going to signal the continuation of transcription. So what is going to happen is, this ribosome, it is going to stall at the two trip codon regions present in this region 1, and this region 2 and 3, they are going to quickly form the loop, that is the anti-terminator loop. And then by doing this, what it is doing is, it is actually preventing the loop formation between 3 and 4. So that means the terminator loop is not going to form, and as a result the RNA polymerase can move ahead with the transcription, and it is going to read the entire structural gene. So this is just like when some relative or father is coming with some sweets or chocolate, and if there are two siblings, definitely the one sibling is going to run faster and make sure that all the chocolates are with that particular person or with that sibling. So with this, what that kid is doing, it is blocking the entry, or it is blocking the other kid from taking the chocolate. So same thing is happening over here. Before the 3 and 4 loop could form, the 2 and 3 is going to form the loop. As a result, that anti-terminator is formed. So this was all about the trp Operon. So what are the key points that you need to remember? Point 1, it works only in the prokaryotes. Secondly, the most important criteria to be followed is the transcription and translation has to happen together or simultaneously. Then the leader mRNA has two trp codons, and the secondary structures, that is what is going to control the attenuation in trp Operon. So these are the things that you need to remember. So now let us try to quickly see it as a comparison between the repression and attenuation. So these are the features over here. So repression, okay, the repressor is going to bind to the operator, and here folding of RNA in the leader sequence, it is going to that is going to take the decision. So the trigger in case of repression is presence of high tryptophan. So that means it knows tryptophan is high, there is no need of trp Operon. And in case of attenuation, there is moderate tryptophan levels. Then result, it is when it is coming to repression, it is going to block the RNA polymerase, sorry. And when it is coming to attenuation, the main criteria is the premature transcriptional stop. It is going to, stop the process abruptly or prematurely. So how can we remember this? Okay, that's a simple trip I'll give you. It is tripipa. Okay, tripa or tripipa. So tryptophan ribosomal movement, that is going to yield the paired loops, and that is how it is going to attenuate the transcription. And one simple way in which you can remember is, suppose you are standing in the very crowded supermarket. So your intention is to quickly get into the queue, finish your billing and go out. So if you are still standing in the queue waiting for, and suppose if your mother has asked you to buy, say, rice, okay, and you're waiting and you don't want to, you don't have patience to stay there longer. So what will you do? You are going to wait, okay, you are waiting in the queue. So suddenly the mother calls you and says, hey, no need to get rice because we have enough at home. So what will you do? You are going to quickly come out, right? Because you don't need to stay there in the supermarket for long, because you were sent to get something, and if it is already there, there is no need to go get it, right? So same way, if there is enough of tryptophan, why your bacteria will need to go ahead with the, put in extra efforts of going through this tryptophan. So same thing happens in case of your trip attenuation. So this is a quick summary. It is a fine-tuned mechanism in which the transcription is going to stop prematurely. So it works after the promoter region. It uses the stem loop RNA to control, and important for gene regulation in bacteria. Attenuation occurs in prokaryotes because the translation and transcription occurs simultaneously. So for more such easy tricks and easy explanations, stay tuned to Cosmic Edutech by Dr. Priyanka and hit the bell button to stay updated. So till then, keep smiling and keep studying. Thank you.